Good morning, everyone. Thank you for coming. So, we E. coli are picky eaters, and we would like to show you how we can use this to our advantage. But before diving in, we would like to present ourselves. We are Marine, Mehdi, and Nathalie. Sorry, technical difficulties. Nantes is a city in the northwest part of France, and this year it was elected the capital of innovation in Europe. Being the first iGEM team from Nantes, we were buzzing in anticipation and bursting with a multitude of ideas. And 1 week, we would go from trying to heat our homes with bacteria to trying to cure depression. But none of these ideas seemed to stick. We just couldn't seem to find the right one.
Until one day, I was sitting across the table from my grandmother, and I watched as she was taking her daily medicine. And I noticed that she stopped for a minute, trying to remember if she had already taken the small oval-shaped pill she was holding in her hand. So I asked her, "Mammy, is it hard to keep track of all the medicine you take every day?" And she told me, "Well, you know, the small pills all have the same shape, the same color, and the same size, so if I don't focus, I sometimes get confused."
So, after Marianne told us about this incident, we went home to our own grandparents and asked them the same question. And actually, many of us get the same answer. But before, I have a quick question for you. Who in this room have found themselves or know someone having to take more than one medicine at the same time? As you can see, it's a pretty common situation. So, at the good scientists we are, we decided to explore the risks of polymedication in scientific articles. And we discovered a key term, medication errors. According to the World Health Organization, medication error caused at least one death every day. And the annual cost of medication error around the world is about $42 billion. 63% of medication error are due to cause undesirable effects. And in France, 40% of medication error are due to an error during the administration phase, meaning this is the phase we should focus on.
All these numbers screamed out for us to go further, so we decided to talk with polymedicated patients, nurses, and doctors to get their point of view on the problem. So we talked to a doctor that remembered tending to an elderly man that had been on a certain medicine that he had to take 3 times a day, as well as sleeping pills. But in fact, he had confused both medicines and was taking 1 pill of a certain medicine and 3 sleeping pills a day. This confusion brought him to the hospital in a semi-coma. So talking to these patients and these professionals in the medical field, we realized there was a real problem. We came up with the idea of a transdermal patch. This patch would be able to deliver different medicines at different doses at different points of time in the day. In doing research on this subject, we also discovered another societal issue that we would like to discuss with you today.
So as Nathalie said, while we were looking for drugs that we could add to our patch, we talked with medical doctor Patricia Lemarchand and data analyst Pascal Kunz, and they told us that posology, which is the study of the dosage of medicine, was mainly based on data from the adult standard men. And this is really a problem because, as you know, there are physiological differences between men and women that affect drug activities. And for example, there are pharmacokinetics and pharmacodynamics, which are the movement and the effect a drug has on the body. So there are also the side effects that differs between men and women. So women really have to be included in these studies. And we talked about this issue with our principal instructor, Émilie Camberlin, and with scientists from our university. And we wondered if that lack of representation of women in those studies was not part of a bigger problem, which is the minor representation of women in science and in STEM especially. So to go further in our research, we interviewed 6 professors from our university, and we designed a survey that was answered by 180 scientists from our area.
So this study really affected our project. We read that medical patch were affected by temperature, and we also found studies that suggest that temperature is directly correlated with the amount of body fat. This means that body temperature is very depending on the part of the body, but also depending on the sex of the person. Taking this into account, we decided that our patch would be on a part of the body where the temperature doesn't vary significantly between men and women, such as forearm. We also looked for differences in pathology between men and women to build our transdermal patch. But Nathalie, how do we get a patch that can deliver the right amount of medicine for the right amount of time, at the same time, each day?
Well, Mehdi, this is where our picky eater E. coli friends come into the picture. We discovered an article by Guy Eidelberg and his team called Hierarchy of Non-Glucose Sugars in E. coli. This article tells us that sugar promoters, or promoters that are associated to the consumption of a sugar, are specifically activated by a specific sugar. For example, P-lactose will be activated by lactose, and P-arabinose will be activated by arabinose, and so on and so forth. Secondly, the article tells us that when the E. coli is placed in a medium containing different sugars, it will consume them in a certain order respecting a hierarchy. So lactose, being the first in the hierarchy, will be consumed first, and then arabinose, which is second, and when all the arabinose is consumed, it will go on to sorbitol, and then finally to ribose, that is last in the hierarchy. We had the pleasure of talking with Guy Eidelberg and engineers and scientists in different fields. And while talking with them, we were able to discover the wide range of possible applications for our tool. And this really made us, we had to make a decision. Did we want to stay in the development of a medical patch, or did we want to try and develop a fundamental tool that would allow gene expression in time with sugar? And this is what we did. We transitioned from a medical patch to a fundamental tool. But Marine, realistically speaking, how can we use this sugar hierarchy to control gene expression in time?
Well, Nathalie, for our project, we use 4 promoters, which are P-lactose, P-sorbitol, P-arabinose, and P-ribose. And by placing a gene of interest under the control of each sugar, we should be able to impact gene expression. Our hypothesis is that by varying the amount of sugar we place in the medium, we should impact the gene expression. Because it's very simple. If you put more sugar in the medium, then it will be consumed in the less amount of time, and thus it will be more available to activate the promoter, and thus to have a gene expression last for longer. So now all we had left to do was to get down to business and build our biological tool, because the aim of our project is to characterize this correlation between gene expression and the amount of sugar. So to do so, we placed a fluorescent reporter gene under the control of our 4 promoters. Under P-lactose, we placed GFP. Under P-arabinose, we placed CFP. Under P-sorbitol, we placed RFP. And the last one, we placed YFP under P-ribose. Then we put this construct into simple insert plasmids in order to characterize the promoters. But then we wanted to lower the size of our tool, because 4 simple inserts is a lot into a bacteria. So we went from 4 simple inserts to 2 double inserts.
So, ladies and gentlemen, this is BioPlug. A new tool that allows the sequential expression of up to 4 genes. And to test our concept, we decided to work with a special strain of E. coli called K12NJ1655 that we transformed with our different constructs. We then cultivated those bacteria in a medium, M9 without any sugar, and according to the experiments we wanted to do, the sugar to be tested is added in the medium.
So now, when we had our constructs, the first question we asked ourselves were, do they even work? And so to test this, to answer this question, we're going to answer it in 2 parts. First, we want to check that the promoters are activated in the presence of their associated sugar. Secondly, we want to see if these promoters respect the sugar hierarchy. So to answer the first part of the question, do the promoters activate in the presence of their associated sugar? We will take the example of P-lactose. So this here, we put the P-lactose in the minimal M9 medium, and we measured the fluorescence in the optical spectrofluorimetry. Here is the curve you can see of this bacteria cultivated in the absence of sugar. We then cultivated these bacteria, transformed with the construct P-lactose, in the presence of the associated sugar lactose, and we obtained this curve. So as you can see, the fluorescence, and therefore the promoter activity, significantly increases in the presence of the associated sugar compared to in the absence of the sugar. From this, we can conclude that this promoter, it functions well. We did the same experiment for our other promoters, And for P arabinose and P ribose, we see the same behavior. So here we have the fluorescence in the absence of sugar. And then when we cultivate this bacteria in the presence of the associated sugar, we obtain these curves. So these two promoters also work. And then, however, for the P sorbitol, we did not observe this behavior, so we were unable to conclude that it functions well. So in our next experiments, we will only present these three functional promoters. So now we can say we have designed three functional sugar promoters. The next step is to see that these promoters respect the sugar hierarchy. We will take the example of P ribose. So we cultivated P ribose in 4 different mediums, each medium containing a different sugar. We did the same thing. We measured the fluorescence and the optical density by spectrophotometry. And as you can see here, P ribose, the activity of the promoter P ribose, is significantly more higher in the presence of ribose than all the other sugars, even if it is activated in the presence of sorbitol and arabinose. This allows us to confirm that P ribose respects the sugar hierarchy.
So now, as we've seen with Nathalie, we know that our constructs work. So what's next? We need to characterize them by studying their activity in time. And to do so, we calculated the fluorescent emission kinetics. And this allowed us to look beyond the accumulation of fluorescent protein. So for P lactose, it enabled us to determine in A, the beginning of activation of our promoters, in B, the beginning of decline of our promoters' activity, and in C, the end of its activation. And from that, we can say that the delay of activation of P lactose is 2.5 hours, and that it stays active for 4.5 hours. And we were able to make the same characterization for the 2 other promoters that were effective, which are P arabinose and P ribose.
So our promoters works, and we have characterized their activity in time. But now we'd like to know if we can if we can control the intensity of promoter activity by varying sugar concentrations. For this, we cultivated our strains of E. coli in the presence of 4 different concentrations of sugar and without sugar for the negative control. And we obtained really interesting results for P arabinose. As you can see on the screen, the intensity of promoter activity varied depending on the concentration of the sugar. However, for the other promoters, we didn't obtain conclusive results, and we think that the problem is coming from the concentration of sugar used that are maybe too high to observe any conclusive differences between on the promoter activity.
So a quick sum up, we have 3 functional promoters. We have been able to characterize their activity in time. And we have seen that P arabinose, we can modulate the intensity of promoter activity with sugar concentration. If you recall, in the beginning we said that we would want to go from simple insert constructs to double insert constructs. So the next logical step is to see that the activity of promoters is not influenced by the change of construct. So what we did is we transformed our bacteria with the double insert constructs, and we cultivated them like before in the absence of sugar and in the presence of their associated sugar. And we obtained this graph for P lactose. So here's P lactose in the double insert construct, and in gray you can see the fluorescence of the bacteria cultivated in the absence of sugar, and in green the fluorescence emitted by the bacteria in the presence of the associated sugar lactose. This allows us to confirm that the promoter is well functioning in the double insert construct. We did the same thing for the 2 other promoters, and we come to the same conclusion. Therefore, we can conclude that our promoters are active in the presence of their associated sugar in the simple insert constructs as well as in the double insert constructs.
So now we have all of this data. What do we do with it? How do we exploit it? Well, during our project, we built a mathematical model that will be able to correlate the amount of sugar in the medium with the duration of promoter activity. And the next step for us now is to implement our latest results with our future ones to make our tool even more predictable and even more precise. And the end goal of this tool is for you to use it. You would input the amount of time you want each promoter to be activated for, so the duration of gene expression you need, and our mathematical model will tell you the amount of each sugar you need to make it happen. It's as simple as that.
So, to sum up, we have 3 fully functional sugar-associated promoters. We have characterized delay, duration, and modulation time of their activity. And we have created a mathematical model that directly links the sugar concentrations with the promoter activity. But what do we have to do now to complete our tool? We first want to redesign a sorbitol promoter that works. Secondly, we would like to redefine saturating concentrations to have a higher specificity when studying that promoter activity. And then we would like to test our different promoters on different conditions such as pH, temperature, and other concentrations of sugar.
So alongside our scientific project, we spent a lot of time and effort trying to open up our project to the grand public. As a lot of other iGEM teams, we realized there's still a lot of fear surrounding the topic of GMOs. So we really had at heart to organize events that would be able to share our side of the story in a fun and accessible way. So we were invited to a local radio station where we presented our project in iGEM for 13,000 listeners. We also held a stand at a national scientific event that welcomed 2,000 visitors. And we presented our project in 2 scientific congresses and 5 events open to the public. We also designed a successful escape game that intertwined elements from synthetic biology, iGEM, and our project. And to escape an old abandoned lab, the players had to understand our project, understand what iGEM was, and how synthetic biology worked. So if you recall, Marine told us in the beginning that we study the position of women in science. From this data, we were able to design a booklet that was aimed to empower students and inspire young people and less young people to pursue careers in science, no matter their gender. And finally, we really had it hard to start a conversation about synthetic biology in a universal language of art. So we organized an art exhibition and we invited professional artists and other iGEM teams to participate in the art exhibition. We would like to take this opportunity to thank them.
So we hope you enjoyed our presentation as much as we enjoyed working on our project. We would like to thank our sponsors and our partners for providing us financially and for believing in our team and in our project. We would like to mention our university, Université de Nantes, and next, Health and Engineering. And of course, the 2 laboratories that welcomed us this summer, which are UFIP and CRCINA. And we want to give the warmest thank you to our PIs and advisors for believing us throughout this whole project and for giving us nothing but unconditional support. You made this adventure so much better for us. So thank you, Laetitia, Émilie, Bernard, Thierry, Dorian, Tiffen, and Vincent. And we would like to mention also our 3 of our team members that could not be present with us today, which are Valentin, Louis, and Paul-Marie. And of course, thank you for taking the time to be here today and for sharing this moment with us.